I'm going to talk about biochemical graph representation and reconstruction using AI and ML techniques. I didn't realize that this is a lab introduction talk, so I gave a generic title. These are some problems we are working on. So in general, the objective of lab is to understand complex networks, where major chunk is biological networks. Also, we are trying to understand man-made and other artificial networks by leveraging the data, which can be a multi-sensor and multi-scale. In terms of biological networks, it can come from different kind of omics data, literature data. Sometimes we also look at different scale in terms of time, mostly. And we use concept of first principles, or what I call it concept of physics, chemistry, underlying biology to understand these networks using tools from control theory, network theory, and learning theory, where we do not want to use blindly data analysis techniques, but also wants to use our insight or knowledge we have gained through hard experiments and our experience in the field of biology and chemistry. So that's what we call it physics-based learning or physics-inspired learning. And our main research interest for achieving this aim is to go through three kind of projects where we look at a process analytical control technology for controlling bioprocesses and understanding them. Second place which we are looking at physically interpretable AI and ML techniques for understanding biological and man-made networks. And today's talk is mainly about how we can bring some of the physics to the biology and develop AI techniques which allows us to get interpretable results if possible, and possible better results than current state-of-art models. And the last is actually, again, the implication of the second one is the network and learning theory for understanding disease and possibly therapeutics in the future. For just to give an introduction, what I call biochemical graphs, it can be called biological networks. I like a word graph because at the end of the day, we sometimes change them to the graph and actually study the graph properties. Metabolic reaction pathways is a biochemical graph for me, signaling pathways, protein-protein interactions, gene-gene regulatory interactions, and interaction between this metabolic protein and gene regulatory interaction, also molecular graphs. There is another called molecule, or at least in the AI ML community, they call it molecular graphs, is molecules and their structure, like you can see here. And these are basically some of the graphs which we want to actually represent for doing some kind of analysis, or sometimes we want to learn them from the data. And today's talk, I'm going to talk about one example where we try to learn the representation of these graphs for property predictions. And another one is actually to build this graph for doing therapeutical intervention in the longer run, or even getting a better insight into disease mechanisms. So when we start with the data, data can be anything. As I told you, it can be literature data, it can be omics data, it can be existing reconstructed networks available to us, it can be molecules. And from using this data, our objectives at the end of the day is to build some network models, which allows us to do some kind of therapeutical intervention in an optimal manner, because many of these networks are needed for getting a physically interpretable therapeutical intervention. So we, unlike completely blind data-driven approaches, we actually have this network models which we use to understand phenotypes, and ultimately use that to actually use that to with the control theory to build optimal therapeutical intervention. For example, in the case of cancerous therapy, we want to know what kind of number of cycles, how often we should give cycles using these models, and measurements which we get from the patient after each cycles. Of course, learning this network itself is a big task before we go to the optimal therapeutic intervention. And today's talk agenda is two-part. First part is to look at how we can actually take the molecular graphs or biochemical graphs to do the property prediction. And one of the important or challenging part here is how to actually develop AI ML algorithms or an approach to get a molecular fingerprinting. Another approach which I'm going to look at, or another problems which I'm going to look at, is a little different from the first one, where instead of representing, I want to reconstruct the graph. And this is again the challenging problem, particularly in context of the last problem which we discussed, where often we use literature data to reconstruct the rough model and then use that rough model to actually refine with the omics data and other data. And the question we are asking that, can we actually reconstruct a biochemical graph or a network from the literature data? And that's what I'm going to discuss, some methodology we have tried to develop in last couple of years. And most of these projects are actually done by undergrads. None of these projects currently is done by a PhD student. Both of these, and that's the reason I actually chose these projects, to show that how wonderful things mostly our undergrads are doing. First problem is related with the property prediction. Motivation for why we are interested in this problem. Motivation is we started as a drug repurposing problem with one of the startup. Of course, that startup has not turned up, but the good thing is that we actually got introduced to an interesting problem in a drug repurposing, and we then realized that it is going beyond drug repurposing, because in a drug discovery, if you see that we start with around 5,000 to 10,000 compounds at a high throughput screening, and out of that 250 compounds will go for preclinical testing, more thorough screening, and out of that 5 compounds will go for clinical trials. And at the end of the day, one compound will be approved by Food and Drug Agency. Here FDA is US FDA or other agencies in the world, appropriate agencies. And you can see that the journey from 10,000 compound to 1,000 compound is not a straightforward. It what they call it in the pharmaceutical industries, blockbusters needs almost $1 billion and 5 to 8 years of research. In a case of drug repurposing, it costs luckily 1/3 of the cost around, and the time is reduced to again 1 to 2 years sometime. So you can see that the molecular property is one of the important aspects, because the experimental screening is time consuming and costly, and the virtual screening is one of the best alternative to not reduce the cost but also time. This cost also has an implication in terms of final cost of drug. So sometimes you wonder why some of the drugs are costly, exorbitantly high. The reason is because the break-even point itself is high. And if we use some of the AI and ML algorithms to reduce this experimental screening, of course, there is no get away without experiments. So let me be clear about it. AI and ML cannot solve experimental problem. What we are trying to do is we are trying to help experimentalists to do targeted experiments, and this is possible through virtual screening, targeted screening. And for that, what we want to do is we want to actually develop AI ML algorithms which predict the properties of interest, and in future possibly generate novel molecules which actually have a desired property, or at least in silico desired property, which can be used later on by experimentalists to create a new or synthesize new molecule of same kind and test on the desired and test in the desired experiments. So First problem, the objective is very simple. Can we actually start with molecular graph and learn molecular fingerprinting using some AI ML algorithms? Earlier, there are simple algorithms, people have used it, and it has actually helped, but still not adequately. And last 4-5 years, this area has become more interesting after the deep learning came into play. And there are interesting algorithms have been developed in last 2 to 3 years to actually represent a molecular graph in terms of molecular fingerprintings. The pipeline is very straightforward. I will start with molecular graph, and I have some algorithm which allows me to get molecular fingerprints. And once I have molecular fingerprints, I can actually use some other algorithm which can will actually allows me to predict the properties. Now the question is, how do we learn this molecular graph representation for property prediction is one question, particularly this aspect altogether. But more important aspect which we want to understand and learn is how can we learn physically meaningful biochemical representation, which not only help us in property prediction, but also in future allow us to generate novel molecules which can exhibit desired properties. And if we do a complete data-driven approach, it may be possible. It may not be possible completely to do that particular task. And of course, first question has been answered by many people, and what we are trying to do is actually try to bring a second question. If we can answer along with the first question, then it would be nice. As I told you that there are many routes to actually learn graph representation. Some people actually start with molecules and take a structure. They actually look at represent this structure as a graph in a graph theory point of view, where each molecule represents some kind of node and each bond represents some kind of edge. And then using that graph, they actually come up with some molecular fingerprint. Similarly, they use some kind of other descriptor like molecular weight, partial pressure, and etc. to again create a molecular fingerprinting. Often, people use both of them together to create molecular fingerprinting and use standard RNN model and CNN model to actually predict the properties. Sometimes they also actually use this RNN and CNN to what they call it molecular embedding, or I call it molecular fingerprinting. Both are same in certain senses. And then again, this fingerprinting they can use to learn, use some other learning algorithm to build a model from fingerprinting to the property and to build that. So the different routes are here, which actually I can see 1, 2, and 3 routes which use structure, smile representation of molecules, descriptors. The only problem is most of this time they use these descriptors are not actually used as a whole, rather than looking at some of this embedding, which is not really physically meaningful. So earlier models which are use RNN and CNN more or less actually doing nothing but molecular embedding, and that leads to a class of neural network, what they call is message passing neural networks. So whatever route I have described here in this slide, RNN, CNN, or machine learning, all of them actually earlier look like that they are actually using this smile string to predict the properties. But all this typically do is some kind of molecular embedding, and then use another machine learning algorithm to predict the properties. Of course, I have a data set which actually have a molecule as well as property together, which is labeled data sets. And around 2015 or 16, there is a publication which actually showed that all these algorithm can be actually can seen as something called message passing neural networks. So what these algorithms do, they actually take a graph, molecular graph or molecules graphic structure, and that represent as a molecular graph. And you can see that each of these molecule atom is actually node here, and the link between them is basically a bond between them. So for example, if there is a between A and B molecule, if there is a bond between them, then there is a age between them. And you can see there is a small circle actually show is called node characteristics. Basically, if you take an atom, atom has a different characteristic, like carbon atom has a characteristic. What is it? Its atomic number. What is its sp3, sp spinning, and other characteristic. You can actually use them to actually represent this atom characteristics. And similarly, for the B, there is a different characteristic. Molecule C is a different characteristic. D, there is a different characteristic. E, and vice versa. Now, with this, I have actually initial setup where I have represented my molecule as a graph. With each node, I have a certain properties associated with each node. And what message passing neural network do is actually pass the message from one node to another node and keep repeating it for a finite amount of time. And with that, actually what they do is there is some kind of, you can think of some functional operation you perform. I'm not going to talk about the math here right now, but you can think that they are passing some kind of functional approximation, keep functional approximation, and you are keep doing functional approximation for finite amount of time, which is done through some kind of graph neural networks. And finally, at the end of the message passing phase, you have a new representation for each of these nodes. And once you have this representation, what you do is you aggregate this representation using aggregate functions, and it's called some kind of readout phase, where you use simple feed-forward neural network to use this as a you can see as the fingerprint of each molecule based on these graph neural networks. So what we have done is we have started with the basic property, and we actually come up with some property of this molecule as its property in this particular atom in this particular molecule. And once this property is achieved, we actually aggregate these properties and create a property of molecule. And once we have that, we have actually at the end of the day, we have fingerprint for this molecule, which is our output. And then we use this output along with the property to build a model which actually predicts the property. The message passing neural network is a first is a set of neural networks. It is not a one neural network. It is basically a set of different methods in that, and typically they use atom information or vertices information. 3 years ago, the same group actually come up with the directed message passing neural network, which is currently state of art. In other words, they showed the performance is quite good, and now any new algorithms which are developed, we have to compare with directed MPNN. So what the changes are from the MPNN to directed MPNN is instead of atom using, they says that the bond has a more important aspect to deal with. So they have actually used message associated with the directed edges on bonds. And these directed edges are constructed based on how particular atom actually interact with another atom. And of course, just for information, although for message passing they associated with bond, but they also use atom features together. So there is a feature for a bond, there is a feature for atom also. The only problem, again, the thing is that does the same. There is a message passing phase, there is a readout phase, little different manner, but the same phases are there. Again, the no physical interpretation of encoding of the molecule at all in the DMPNN. It performs well, of course, but if I want to actually do generate a new molecule, I have to go to completely new training and new way of solving the problem. So what we thought is, can we actually bring some physical meaning to encode the molecules. And what we thought when we discussed about it, we thought that, okay, functional group is something which many people actually discuss. I think Karthik has a nice paper about that, where they look at reaction property using reaction code or the functional group related properties, which is nothing but a substructure of molecules that causes the molecule's characteristic, particularly in chemical reactions. And group of atoms is a actually functional group consists of a group of atom in a molecule, and they actually, again, we thought that attribute to distinct chemical properties. And now the question we are asking, there are around 500, 600 functional groups are available in the literature, but we say, okay, let's not worry about them. Can I actually take a molecule and identify functional groups in the molecules? And second question we ask, if I actually identify a functional group in the molecule, how should I actually use this functional group to encode the molecules? And that leads to a functional substructure representation, which we have developed in last 1.5 years, where what we did is, rather than using molecular graph, we actually use SMILE, or which is called Simplified Molecular Input Line Entry System, which represents the molecular graph as a simple sentence or sequence of, you can say, molecular arrangement like this. It's a line representation, and it is, you can think it as like a sentence representation of molecule. And what we are interested in is in this particular sentence is to find out what are important repeating words. Okay, so in the language, when you talk about, some of the words are not important, like the, a, sometimes, particularly when you look at large corpus. But some of the words, like which are context specific, or like activates, transforms, or molecule, or let's say inhibits, those kind of those are words which, if in certain context, is very useful. So what we are looking here is, can we identify substructure or common substructure across different molecules which are repeating frequently? Okay. And the question we ask, given SMILES, can we find these common substructures in an easy manner? Of course, you can do the same thing with a molecular graph, where you can find substructures using graph mining algorithms. But they are a little bit complicated. We thought that why can't we use SMILE and see, can we harness functional group properties just by using SMILES? And what we did is, we used chemical sequential pattern mining algorithm, which has been published a couple of years ago, 2019, by one of the group, which actually was trying to solve some different problem in a context of NLP, which natural language processing, and they actually use for certain small problem, same idea, where they actually use pattern algorithms to find out common substructure in a sentence or in a molecules. So we use that, and what we did is, we went to a database which has around 2 million molecule structures in a SMILE format. We applied chemical sequential pattern mining algorithms and found out a set of frequently happening substructures. Of course, there are huge number of substructures. So what we have done is, we said that, okay, we'll take a substructure which has at least repeats 10,000 times or 5,000 times. And with that, we actually got around 2,500 odd substructure which are repeating frequently in these 2 million molecules. Then what we did is, we actually used this substructure to actually represent a chemical structure or a SMILE of a particular molecule. For example, in this particular case, this is a SMILE, and we use and see that, okay, this is a repeating substructure, this is a repeating substructure, and we see that which one is actually repeating in the substructure which we think are really important substructure, or you can think this as a vocabulary. So I have a sentence which I construct from a set of vocabularies. I have created a vocabulary by reverse engineering the sentences, and this is the vocabulary I think a molecule is representative of. And what I do is, what we call it in machine learning is called one hot encoding. So I have a huge 2500 dimension big vector, and I see whether this particular smile consists of what substructures corresponding to this 2500 substructure I have actually discovered in my data here. Then if the substructure is present, I actually encode as a 1. If substructure is not present, I encode as a 0. And at the end of the day, what I will have is, I have an input representation of my smile string where the substructure in that particular molecule is present out of those 2500 substructure I have discovered or my vocabulary. If it's out of vocabulary, I do not consider at all. And we never found, very few were out of vocabulary, like around hundreds or 200s. Rest of the time, we actually found a molecule which has a substructure and we represent as a 1 or 0. And this is what I get as my input representation. Then what I will do is, I will take this substructure and actually encode, decoder, which is autoencoder I will use, to actually represent, learn this representation. Autoencoder you can think of is basically a function where if x is equal to f of x. Now someone say, oh, f is equal to f of x, what you learn? So what I learn here is basically a latent representation or that function which actually encode this representation in an even more parsimonious manner. And this latent representation actually can be decoded, and I can get back my input representation back. And this is called autoencoder in a machine learning. And we actually use them to learn this latent variable and use this latent variable and the property, which is label data, to build a regression model. And using this latent variable and the property, we build a regression model. And whenever you give me new chemical structure, I will pass through this encoder, get the latent representation for that particular chemical structure, and use the model which I have built to predict the property. The interesting fact here is this particular input representation is a kind of representation of molecules, and there is a way in a generative model I can go from the predict property to this one. And before, I mean, that is the future what we are going to do it, where we can actually use generative models. And here the interesting aspect is that I know what kind of functional group I need if I can do the generative model at the end of the day for a particular kind of property of interest. And that's where the difference lies with some of the algorithms which are there. Now the question is, does this particular approach do the better job? So what we have done is, we have actually tested this approach on set of data set. I mean, we have tested on more data sets. I'm just showing you some of them. And we can see that in some of the cases, it is behaving better than the DMPNN. Sometimes it is performing bad, but not really bad. Sometimes it's performing as good as it is. And we found that FSR works quite well in many cases and actually outsmart DMPNN. Currently, we are still working on the FSR. We are trying to improve it. And let me summarize what we have done is that we have started with physically interpretable or chemistry interpretable representation of the molecule, which allows us to encode the molecule using the functional substructure representation. And the encoding is so far as good as DMPNN. It has a physical meaning because we know what kind of substructure are there, which again is going to be useful when it comes to developing generative models where I want to build, no, I want to construct a new molecules given the property. The other things which I wanted to point out in this particular aspect is that we have not actually used many other descriptors which are used by DMPNN. So DMPNN use a lot of molecule-specific structure information like molecular weight, the orbit, orbital structure, atom, and other aspect. There are bond, what kind of bonds are there. In FSR, we are not using anything. We are just using a simple molecule as an input. We are not characterizing what are this particular CO is there, what is its property. No, we are not doing anything. We are just, it's a very simple approach. And you can see without using a less number of structure, it sometimes behaves better and often behaves as good as the DMPNN. Other things which we have not done is we have actually built this model for a case-by-case basis. In other words, when I did this training at this end, not this end, this is we have done for all of them, but this training is done for each data set, which is I did for a data set for this data set, this data set, this data set individually. And what we are currently doing is we are trying to do pre-training. In other words, we are trying to do training of our encoder decoder using 2 million molecules in a one go. It's called pre-trained models, and it has its own advantages where we can actually get a better representation than we use a subset of molecules. So these are two future outlooks we are looking for, and hopefully by end of this year we have a not end of this academic year, we have a better idea about what happens to pre-training. Of course, we have also compared this particular aspect with the known functional groups, and surprisingly, it's behaving very poorly in many cases. Almost 80% cases, the functional group which our chemistry has actually given to us are behaving poorly when we use FSR representation. And it again asks the question that is the current functional groups which are there screened by chemists are adequate, or should we go back and see the definition of functional substructure we computed and compare them and see how far they are from, how many are repeating. So those analyses currently we are doing, and maybe we go back to the chemist and ask what unknown functional substructure represent and does that have meaning. So this way we can actually add more functional group which chemists should look for in the future. Okay, so I think I have a next 10 minutes to 15 minutes. So let me talk about another problem very briefly. I'll skip some slides in the interest of time. But this is another interesting problem which we wanted to solve. And partially, I think, thanks to some of colleagues, including Karthik, about it over some discussion. The question here is following, that we have a set of publications. And a publication is a lot of biological knowledge. Many people have actually spent years and years in the lab to figure it out. What are the interactions? What are protein interact with other protein through cumbersome experiments? Similarly, for example, we deal with constrained models where metabolic reaction networks model has to be created, where we actually create using Recon model plus use omics data. Still, we go back to the literature and try to see for the particular context what more interaction we have to add. And this again is a cumbersome process. So the question we have asked is that, can we make this process automatically, or at least for a new, completely new problem, new context, can we reconstruct a rough network using the literature data? So the objective here is simple. We want to automatically reconstruct a biological network in a particular context from the literature data or published data using either set of abstract or complete text articles. And this problem we try to solve through three tasks. First task is we want to first, you can think this, we want to actually pseudo represent the human reasoning, where we actually read and okay, you know set of metabolites when you read in the sentence, what are the gene, which are the proteins, which we call name entities. But computer does not know that. Correct? So we have to first learn somehow how name entity recognition task, where computer should learn what are participating name entities in given text. Once we know that, what we are interested in learning that in some sentence you have a protein 1 interact with protein 2, then we want to learn that okay, protein 1 and protein 2 are interacting. This is in a sentence. It's very straightforward to say when we read it, because human intelligence is or training is different. But again, we want this training to we want to train algorithm which actually can extract the relations in the sentence, or more importantly, desired relationship from the set of sentences. So the second, once we find the entities, we want to know, are there any relationships present in between this entity in this particular sentence. And the last task is, once we have this one, we want to represent this relationship as a network. There are a bunch of work has been done in NER and RE task. It's quite widely been done. It's of 1999, 1998. These particular articles are more about expert system rather than what I call it machine learning systems, where they have a hard-coded, they have hardcore rules to actually extract the data. It worked well at the time, but as the corpus increases, these rules become redundant sometime, or sometime they cannot actually accurately predict the relationship in the sentence. And in the last couple of years, pre-trained models in a language has provided a huge impetus to many research in natural language processing. In this particular case, we are interested in looking at biological literature, use this pre-trained model and adapt to the biological literature for doing the task of network reconstruction from this data. And just a couple of years ago, first benchmark data set has been produced, very small data set, but still for NER and RE task, not for a graph reconstruction task. Graph reconstruction from the sentence is still what we call it novel problem. We have sometimes difficulty to explain to the community. So what we are doing is very simple. We take a text and we use pre-trained model. Pre-trained models are the model which has been trained on huge corpus, which is a neural network model. So you might have read somewhere article that now there are language models who can write articles as good as the human being. Those are actually pre-trained models. And here we are using a pre-trained model which is publicly made available by Google called BERT. So what we did is we use pre-trained model, but we actually tweak some of the pre-trained model to train on our text. So pre-trained model has been already trained on huge corpus, but we add something called transfer learning we do, and by adding a new layers to it and train it again. And using that, we actually build any NER entities which actually can, given a sentence, it can find it out what are the name entities in that sentence. Once we can do that, once we have name entity, what we have done is we have built another model which actually extract the relationships from this particular given text and the entities. So we have a sentence and the entities. Now it is a kind of extended sentence you can think of, which this is the entities are in this sentence, and this is a sentence. Can you tell me are there any relationship between these two entities or these three entities? And we actually build another model which actually extracts the entities, and once we have relationships, we construct the knowledge graph. We call it knowledge graph now because this is what the community calls it, or it is basically nothing but our biological network which tells you different entities which are protein or gene or disease kind of formulation. As I told you, we actually did it using BERT, and there is another specialized called BioBERT. The difference between BERT and BioBERT is BioBERT is actually trained further on biological data. So it is more towards the biological data, trained further on the biological data, so it can handle the biological data or the example which we are interested in. BERT is a very generic tool, but we found out that BERT and BioBERT more or less behaves same when it comes to the task which we are interested in. The main aspect is we have to actually tokenize the sentence, and there are different ways of tokenizing the sentence. In a interest of time, I'm not going to go to that detail. So what we do is we actually take a sentence, we use the BERT and one more layer, which is what we call softmax layer, to predict given sentence whether what are the entities in the sentence. So for example, if in the sentence there are gene disease relationship extraction, we want to know given sentence what are the gene and what are the diseases in that particular sentence, which is, we call it entities. So cancer is a disease, which is entity. GTTA1 is gene, which is also entity. Of course, this sentence is not this simple. This is just for illustration purpose. Sentences are more complicated the way we scientists typically write, or researchers typically write. And then again, what we do is we build a model. And similarly, we do for once we have what you call it, entities, we go for a RE task, and then we again build a model. These are classifier model, which actually predicts whether there is a relationship between two models or not. And here, the first pre-trained model is actually only provide whether there is a entities in the sentence. What are the entities in the sentences? And the second model will actually take these entities along with the sentence and try to predict whether there is a relationship between them or not. So we did different kind of pre-training also to try to create label data for RE task based on the NER task. And there are different ways actually. If you input, it provides a different solution. That's a very interesting aspect. And but we figured out this optimal way of doing it. And once we did it, at the end, what we have to do is we have to take these relationships and figure it out whether there is a relation after relation extraction. We have to figure it out whether there is a relation between two nodes or not. So this is a part of small complete graph. I'm not showing complete graph is very big, and you will not find anything. So this is a complete graph, and what is interesting aspect here is that some of the relationships which we have predicted are predicted well. Some of the relations we could not predict. By the way, we could not predict there is a relation between SPN and TNF alpha, and there is a ARTP1, and that all is actually has a relationship. But interestingly, we actually predicted one new entity which has a relationship. And this is again interesting sometimes, because this relationship does not exist in the original graph or original corpus. At least this is a trained corpus, so human expert has actually not shown this as a relationship. So what we are asking here is that is this really a relationship, or it is a false positive, or what you call it's a falsely represented relationship. And this can be actually go and this provides a new knowledge. Sometime it can be just a false positive. So for an experimentalist, I have to go and show that, okay, EH and the NFP, do they have relationship or not, or are they interact with each other or not? And that might be possible they interact, but the human expert has not actually figured out in original graph there is a relationship. So we got a decent result with this. For NER and RE task, we have done really well. In a graph, our F scores are okay with both of them. Here in particularly BioBuds in this particular data, because this is the only data where we have a complete knowledge. And this actually little bit better than the benchmark data algorithms people have used it. Now we have actually done for many other data. We have done for GAD data. We have done for GP NLP data, and now we are actually trying to see what is the performance. Performance is as decent as this one, and we try to improve it by actually creating what we call it XBERT, which actually allows you to even train further your BERT towards particular field-related vocabulary. A BioBERT is not BioBERT only train the BERT. Here actually you can extend BERT in an XBERT where you have more neural networks and try to solve, try to train that using the biological data. Some of the important learning here we have is we have a significantly improved results in NER and RE task compared to state of some of the algorithms available for NER and RE task for these data sets in the literature. With the increase in training data, the results set better, and that's what we saw. We saw that BioBERT are better than BERT in several cases, but when we actually put more cases, now both are look same, which I have not shown those results here right now because I was not sure whether this conclusion is right. So I'm right now keeping this as a conclusion, when maybe we'll revisit this conclusion as we train on more and more data sets with the same approach. Domain-specific vocabulary is more important in a pre-trained models, and can we actually tune this pre-trained model to bring domain-specific vocabulary and improve their predictability to go towards 70-80%. By the way, that is really good. And in future, we want to actually do some of this analysis like extensive vocabulary training method. We are currently actually trying to construct a network for ovarian cancer and see whether we have tried to do for manually annotated data set we have created. Now we are trying to do that because of one of our PhD students. Another aspect, which is graph reconstruction on various data set, is also key part, and how to actually compare is another aspect which we are looking at. And there are a lot of confusion in the literature itself, so we are trying to solve it out how we should compare our model with some of the existing model. So I would like to thank with that. To conclude, as far as our lab concerned, we work on at the interface of biology, control theory, and learning theory. And our objective, as I told you, is to understand complex processes using theoretical and experimental tools sometime. And this work is jointly done with now it's moved from IBSC. IBSC is now Integrative Biology and Systems Medicine, and Robert Bosch Center for Data Science. And thank you, these 4 students, Gunshekar and Abhishek are dual degree data science students who did most of this work. Joe and Hari Govind are actually doing and improving some of this work and doing more analysis on the work. And thanks RBC Desai, IIT Madras, and IBSC for funding. Thank you very much. If you have any questions, please ask.
I have a question. Can I go?
Yeah, please.
Okay. So I was wondering, you spoke about methods to infer biological networks from literature, right?
Yeah.
How do these methods generally perform against similar methods that infer network connections from correlations in transcriptomic data or proteomic data? Like, do they do better?
No. Right now, no.
I see.
So,
Okay.
Satvik, the idea here is to add those uh, methods rather than constructing a complete new method, because those
Right,
methods
okay.
are mostly be done based on what is available data, correct?
Okay.
Here, actually, uh, researchers have actually already performed experiments at a small scale level, established there are relations, there are interactions, and they have mentioned in somewhere in the literature that gene 1 interact
Okay.
with gene 2, correct? And
Yeah.
this is been thoroughly analyzed. And so if I have to give a what I call it link some probability, I would give a more weight to the model developed by this one. But it is not complete, because as you see, the efficiency is not yet that high. While the omics data or transcriptomics data you are talking about has a is developed for particular context, and there so far the results will be as little better according to what I have seen than what we have done. We try to achieve that if possible, but most of the time those analyses done for a new context, not the available context. If people already know there is an interaction, then there is no point of doing it, those experiments. So to answer your question, currently no, these methods are not doing it. The only good thing about this method is that I can provide it as an added advantage. As I told you earlier, that I'm not interested in constructing complete network, because I'm not going to do it, because I need a more data to construct a complete interaction between different entities. For that, I need omics data. Here, the idea is to start somewhere where what kind of experiment we want to do, what are the gaps we want to fill using the omics data can be done here.
Okay.
Okay.
I have a small follow-up question.
Yes.
So, when you're dealing with omics data, sometimes it's hard to try and infer the directionality of interactions, right?
Yes. Yes.
That is something you could potentially do with NLPs, is it?
Yeah, it is possible. Yeah. Currently, we
Okay.
are not doing it, but yeah, it is possible. Even directionality like activate, inhibit, yeah, that is possible.
Yeah.
Yeah.
Okay. Very cool. Thank you.
Just to add that there are omics data where because of missing, another problem with omics data is a missing value, and those kind of, and the quality of data is still poor so far. So in that case, again, this can help to actually give a weight. If you found a relationship between two this, then you can, at least this can help to say whether these relationships indeed exist in the literature or not.
Okay. Thank you. The talk is very nice.
Any other questions?
How come the students are very silent here? Also, I thought at least in this group they'll ask more questions.
Or I will pick the students. I know half of them.
You should pick students and ask questions. Okay, so Nirav, I have like one sort of very broad question. Right? See, you're doing lots of learning on graphs and stuff like that, right? And in this context, I've always been fascinated by what Ravi had done long time ago. So this was using message passing and things like that. I think it was something called like collective classification and things like that, right? I'm just wondering if basically using the network information, right, in terms of, it could be even for like BioBert or something like that, right? So the kind of entities that connect to each other, right? The patterns of connectivity between different kinds of entities and things like that. Can that be exploited to further inform your task and things like that?
Yeah, that is possible. That's, people are trying to do it nowadays actually. Not, but what you are saying is using the network itself as a data, correct? That's what.
Also. right? I mean, over and above what you already have. For example, I think like Ravi's example was they were classifying active sites and non-active sites in proteins or something like that.
Okay.
But if you classified one node as active, then it alters the the probability of that label for the neighboring nodes.
Okay. Oh, I see. Oh, okay. What you're saying.
Okay, so basically, you collectively classify them. You don't like independently classify. See, the independent classification is what we are all like typically used to.
Yeah.
But by virtue of their networking, there is some more information that is there, and I mean, we tried this on some other thing, and it was like complicated and so on. But I just like the idea very much. Right, because this notion of the network structure has a lot of story to tell you.
Yeah. So okay, I understood. So this, I think you think this way. In fact, currently we are discussing about. We actually revisited your old paper with that reaction prediction one, correct? If you recall. That paper where you actually try to find it out subgraph or reaction center.
Right.
So this FSR is doing something similar in some sense. If you think that what is it doing is it actually clustering this completely this node. Means if I say that this is a common substructure, then I'm actually taking complete one node plus another node as a what you call it, classifying it as a together as a have a certain properties. Correct? That's what you are trying to say. Yeah, so we are also investigating right now to see that can we do it for generically on a more cases where take us this one and then say that whether this particular substructure and see that whether we should include more molecules or not, or whether this let's say here if I take this substructure alone, and now question is that whether should I extend my substructure to here or not is a question we sometimes ask. And that's what you are saying, that if I classify these three nodes as has a certain same property, by just being adjacent to it, you should be in that in the part of that node. Is that what you are saying, right?
Yeah. See, I'm not sure how to sort of adapt it to this thing, but yeah. So the general idea is basically trying to use see here, of course, here you're treating every molecule as a graph, right?
Yes.
Yeah. So in that, there is the complexity is not like so high for learning from network structure. But I think in some other scenarios, I'm sure maybe we can learn something here if you. For example, we embedded all these network structures onto a graph and did some pathfinding on that and things like that later on. So at that point, maybe there's something. But I was just throwing it up as a general idea because I think it seems to have a lot of promise and it seems to be highly under studied.
Okay, so you are saying that if I have protein 1, protein 2, and protein 3, and if I have a same process they are together, is protein 3 is interacting with protein 1, protein 3 is interacting with protein 2, then by this definition you want to say protein 1 and protein 2 interact, something like that. Is that what
could be, right?
Yeah,
so could be. So something like that. So basically this network structure comes into play.
Okay. That can be done. Yeah, that can be done. Yeah. We have to think.
And you also see, so we had this net genes and all that, So we just take all the centrality measures and things like that, and we are able to predict essentiality and things like that. So just the network position, by virtue of a protein, an object's position in the network, it has several properties. So can those be exploited somewhere? Yeah,
but then that has to be exploited. You are saying is exploited as a part of training, or it's exploited after post training. That can be done post probably, or you think about
possibly, yeah. So you can probably it could be one more way to score your thing, right? So if it satisfies some prior network assumptions or something like expected network architectures or something like that, even. Okay. I don't know.
Yeah. No. And this is a good question because currently we just started to work on this reaction prediction problem. So given a set of reactant, we want to predict what are possible molecule it can generate. So in that case, these kind of things are quite useful, because basically you are saying by association, maybe this kind of characteristic will be useful to predict.
Something, yeah.
Yeah, that this particular bond should exist because there is some node clustering happening together.
Yeah.
Okay. Yeah, that's possible. We should think of it. Yeah.